Hello student, how are you? I hope you all are happy, healthy and fine at home. Do you remember what were we discussing yesterday? I think we were discussing about the synthesis of glycosides. Among that synthesis of glycoside, the last point we have discussed, alkylation of reducing sugar. What do you mean by the alkylation of the reducing sugar? How alkylation present there and how it can be, or how to recognize whether that sugar or whether that derivative, carbohydrate derivative is exhibit the reducing property or the non-reducing property that depend upon the structure. Those structure exhibit shows hemiacetal derivative, that type of the organic compound generally produce or shows positive toluene test as well as filling test, and therefore that type of the compound are responsible for reducing sugar, and those structure does not contain hemiacetal part in their compound. They shows negative test, they do not show positive test with Toulen's reagent and the filling solution, and that type of the sugar unit generally termed as a non-reducing sugar. So I think up to this we have discussed this point. So today onward, one important point as far as this syllabus is concerned of carbohydrate chemistry. Carbohydrate chemistry, as we know that it is a polyhydroxy optically active containing formyl group and the ketonic group. That type of the compound is called as a carbohydrates, or other way around we can define carbohydrates are polyhydroxy aldehyde or ketone, or a compound gives rise to a sugar unit on its hydrolysis is known as a carbohydrates. So now if you see this point here written, that is a last point, C is a synthesis of disaccharide, trisaccharide, polysaccharides. So what do you mean by that disaccharide, trisaccharide or the polysaccharides? If you remember the classification of the carbohydrates are mainly, the other alternative name of that carbohydrate is a saccharide, and that carbohydrate mainly classified into three types: monosaccharides, oligosaccharides and polysaccharide. These are the main classification of that carbohydrates. Monosaccharide do not hydrolyze, it is a single unit, and the simplest compound is a D-glucose. Why it is termed as a D-glucose? That is we have learned with the suitable example. Second one is a oligosaccharide, and as we know that oligosaccharide further divided into disaccharide, trisaccharide, tetrasaccharide, and oligosaccharide means this is made up of by 2 to 10 sugar unit combined together. That type of the Carbohydrates are termed as oligosaccharide. Disaccharide means two sugar unit combined together. The resulting compound form is called as a disaccharide, or on hydrolysis. Generally, hydrolysis are carried out in presence of dilute acid or specific enzyme gives rise to two sugar unit. So that type of the compound are termed as disaccharide. The word itself suggests that disaccharide means two. And another one is a trisaccharide. On hydrolysis of that trisaccharide with dilute acid or specific enzyme gives rise to 3 units of carbohydrates. So that type of the thing is called as a trisaccharides. And tetrasaccharide, the name, the word itself suggests that tetra means 4 sugar unit are involved in that, and that's why it is termed as a tetrasaccharide. After the hydrolysis of tetrasaccharide in presence of dilute acid or specific enzyme gives rise to 4 sugar unit, maybe as a same or different unit, that are known as tetrasaccharide. And polysaccharides, more than 10 sugar unit is combined together and formed as a complex compound. That type of the saccharide are known as a polysaccharide. Poly, the word itself suggests that number of sugar unit combined together or involved in that is known as a polysaccharide. So, but at this level, let us see preliminary synthesis of disaccharide, trisaccharide and the polysaccharide. Little bit introduction regarding as a disaccharide. There are commonly 3 observed disaccharides are well-known disaccharides are sucrose, maltose and the lactose. If you remember, these are the 3 commonly naturally occurring disaccharides, and that sucrose, if you find out whether it shows reducing properties or no, whether it is a reducing sugar or non-reducing sugar, how do we recognize or how do we identify? And if you know the structure of that sucrose carbohydrate, and as we know that sucrose are made up by or the sucrose involving the 2 units, one is a D-glucose and another one is a D-fructose. With the combination of D-glucose and D-fructose, there is a formation of glycosidic linkage is present. Generally alpha beta 1,2 glycosidic linkage is found in sucrose, and if you observe the structure of sucrose, you will find that there is no hemiacetal structure present, and because the glucose, the anomeric hydroxyl group involved in the formation of glycosidic bond or the glycosidic linkage, so there the hemiacetal structure is not present in the sucrose, and therefore sucrose is a non-reducing sugar. That is one of the important notable point you must remember as far as that disaccharide of that sucrose is concerned. Next commonly observed disaccharide is a maltose. After the hydrolysis, that maltose gives rise to the compound of 2 units of glucose. In the same D-glucose, this maltose is made up of by 2 sugar units from the D-glucose, and one of the D-glucose is utilizing its anomeric hydroxy group for there is a elimination of water molecule takes place, and the glycosidic linkage or the bond is formed. The resulting compound obtained is known as a maltose, and that maltose, if you observe the structure of maltose carefully, you will find that there is a hemiacetal group is present, because one of the glucose unit utilize its anomeric hydroxy group for the formation of the glycosidic linkage, and there is a one hemiacetal or the anomeric hydroxyl group is present of the second unit of second sugar unit, and therefore, and as we know that the hemiacetal group present over there at an equilibrium, that ring get opened and it can act just like as a free formyl group, and once the free formyl group is formed, it immediately react with the Toulin's reagent and it gives a positive test with Toulin's reagent. Similarly, it can react with the filling solution, that compound, that aldehyde group is get converted into as a carboxylic group, salt of that carboxylic group, and this also shows the positive test of filling solution, and therefore maltose is a reducing sugar. Then third example is a lactose. As we know that lactose is present in the milk, and it is made up of by 2 sugar unit. One is a galactose and another one is a D-glucose. When they combine together with the elimination of water molecule, and there is a formation of glycosidic linkage, and the resulting compound obtained is known as lactose. And if you observe the structure of lactose, here also one of the sugar unit utilize its anomeric hydroxyl group for the glycosidic bond formation, or the glycosidic linkage is formed, and on the other unit, second sugar unit has hemiacetal groups present in it, and once the hemiacetal group is present, that type of the carbohydrate or the sugar unit shows positive Toulin test as well as positive filling solution test. Correspondingly, we or respectively that aldehyde group is converted into salt of that respective metals, and that is a thing. And another one, trisaccharide, means it is made up of by the 3 sugar unit together, and then observe there whether they exhibit or whether they shows hemiacetal structure in their compound or not. If hemiacetal groups present in their structure, that compound gives rise to the positive that hemiacetal group is responsible for exhibiting or showing positive test of filling solution as well as Toulin's reagent. And if you see the polysaccharides, poly means many sugar unit combined together via glycosidic linkage. If you see the cellulose is generally, and again polysaccharide, there are three commonly polysaccharides are observed. Number 1 is a starch, number 2 is a cellulose, and number 3 is a glycogen. And that starch is a main constituent of carbohydrates. It is stored of carbohydrates. Starch you can everybody knows as a potatoes containing as a starch, and that is a storage of carbohydrate. Then cellulose, it is a main constituent of the cell wall of the plants present in cotton or woods. Third one is a glycogen, it is also as a one major constituent. It is a storage constituent of animals, where it is found in muscles, liver and the brain. And if you observe the structure of the cellulose, there is a beta-1,4 glycosidic linkage is observed, and that due to the specific enzyme only animals has, so that they can digest easily, but the human being is not having that type of the specific enzyme, is not present in human's body, and therefore they cannot digest, because there is a beta linkage is a very hard to break that glycosidic linkage, and therefore these are the sum of the introductory part we have seen over here. That the introductory part also we have seen, but the focusing point or main point over here is a stereoselective synthesis of beta-amanocytes. With this point we are now discussing stereoselective synthesis of beta-amanocytes. Mannose, you know that, and that mannose derivative, why we are getting as a beta, and generally there is a mixture of the products are formed. One is a alpha and another one is a beta, but mainly stereoselective, these words clearly indicate that whenever there is a formation of the product, majority of the product are beta product, and therefore it is termed as a beta-amanocytes. And whatever in a previous point we have discussed as a synthesis of glycosides, similarly here is a stereoselective synthesis of the beta-amanocyte. How we can get as only this beta-amanocytes? Let's talk little bit about it. Beta-D mannopyrronyl residues are frequently found in oligosaccharide, and as I already told you that oligosaccharide means what? Oligosaccharides are further divided into three types. Just now I told you that, and that oligosaccharide and glycoconjugates in living system, glycoconjugates, and this unit is omnipresent everywhere in N-linked glycoproteins that contain a pentasaccharide core 1 in which the beta-linked mannose residue can carry additional sugar 1 that is represented in the scheme number 1. Let's see over here is alpha D mannocyte. In short, I am showing here 1 to 6. That is the scheme number 1 I am going to represent over here. Now, here is a beta D mannocyte, and in the bracket 1 to 4, that is the linkage is shown over here. Then beta D GLC N-acetyl, then in a bracket 1:4, that is shown by linkage. Then beta D, there is again GLC NaC, and then in bracket known as 1ASN. This is a compound number 1. This is a compound number 1 shown over here, and this way here alpha D mannocyte from 1 to 3. These are the 3 sugar unit combined together, and from above 6 combined, and we are getting as a compound number 1 that is represented in a scheme number 1st. So beta mannopyranose moieties are often part of the O-specific polysaccharides, and in short it is written as OSPS. OSPS means O-specific polysaccharides. OSPS, O-specific polysaccharide of a gram positive. Here some of the words are coming from the biological point of view, so you should remember gram positive or gram negative, that type of the bacteria, as we know that gram negative bacteria and of bacterial capsular and exopolysaccharides. Beta-linked mannopyranose is also a common residue in plant polysaccharides. Further example of the occurrence of the beta-mannosidic linkage are the glycosphingolipids in wheat flour, and in the freshwater bivalve Hyripesis scaligeli. All these words are from biology. Interestingly, this residue is absent from mammalian glycolipids. Cell wall phosphomammols of yeast contain linear chains of up to 14 beta-1,2 linked mannose residues. Beta-mannopyranose unit also occur in secreted small molecules. Examples include the phospholipase inhibitor, caloporoside, and free oligosaccharide detected in a patient with mannosidosis and isolated from ripening tomatoes. The related beta-linked 2-acetamido-2-deoxy-D-mannopyranose also frequently occurs in OSPS and in capsular polysaccharides. The corresponding mannuronic acid is present in the ester bacterial common antigen and in other bacterial polysaccharides. The occurrence of the beta-amanosyl unit in many biological systems creates a biological rationale for its synthesis, whereas simple methyl and phenyl beta-D-mannopyranoside have been known for many years. The synthesis of complex oligosaccharides containing these units is still a challenge for two reasons. Reason number one, listen carefully. First, the anomeric effect facilitates the formation of the 1,2-trans mannopyranosyl alpha linkage. Thus, it disfavors the beta linkage, and second, the 1,2-cis arrangement of the equatorial glycogen and the axial functionality at C2 in beta-mannopyranosides harbors repulsive steric effects. Glycosyl donors with a participating group at O2 and halide ion catalytic conditions promote the exclusive formation of alpha mannocytes, and these are the some of the introductory part regarding that stereoselective synthesis of beta mannocytes. So here we have seen. In the next lecture we are going to discuss some of the scheme regarding chemical methods. These are the biological method we have seen: glycosylation with mannosyl donor, mannocylation using insoluble promoter. So that type of the thing, how it can be act, and we are going to synthesize different disaccharides as well as trisaccharide by keeping the point of stereoselective synthesis of beta mannocytes. So I think with this, let us stop today here. Remaining we will see in the next lecture. Thank you. Have a nice day.